Hello, good evening to all. Visible, audible.
हाँ
Yes,
सर।
sir.
Okay. So this is first class of Unit 2. Unit 1 is completed. Notes are given. Start to read and Unit 1 exams conduct soon. And the date is also given. 30th September. Unit 2. Diversity of microbial world. Diversity of microbial world. Diversity of microbial world has part A and part B. Part A is all about the systems of classification. Systems of classification, and part B is description of bacteria, protozoa. Description means morphology, importance role, economic purpose, reproduction style, mode of nutrition, etc. Description of bacteria, then protozoa, fungi, and protist. So today we start part A. Part A, system of classification. System of classification. It is clear that microbes are highly diverse. Yes or no? Microbes are highly diverse in nature in terms of they can be unicellular, they can be multicellular, they can be prokaryote, and some are eukaryote. Number two, some microbes are acellular, some are cellular, a majority are cellular, some are acellular. Can you give examples some acellular microbes present in our living world? Acellular. That means no cell structure, proper cell structures are there.
Virus.
Virus, virus, other viroids, prions. These are all acellular, not properly structured cells are present. Okay, and majority are cellular. That means bacteria, cellular structure, fungi, protozoa, etc. All are cellular. But some microbes are also acellular in nature, and they also diverse in terms of mode of nutrition. Mode of nutrition, because microbes can show wide diversity of nutrition. They can be autotrophic, they can be heterotrophic, and previous day class soil from the soil microbiology we know the autotrophic organism can be of two types. One is photoautotrophs, other is chemoautotrophs. So autotrophic microbes are also be two types: photoautotrophs Who discovered the concept of chemoautotrophs? Previous day class question. Who discovered the concept of chemoautotrophy?
Sir
Which
Paul
scientist?
Ehrhart.
Paul Ehrlich. No. Concept of chemoautotrophs. Soil microbiology. Establishment scientist contribution.
Sergey Vinogradsky.
উইনোগ্রাস্কি উইনোগ্রাস্কি কলম যেটা প্রিপেয়ার করলো উইনোগ্রাস্কি সার্জে উইনোগ্রাস্কি concept give the concept of chemoautotrophs. That means he showed that some microbes can utilize simple inorganic material for the source of energy. Photoautotrophs, they need that energy of light. Are they using simple inorganic material like iron, sulfur, etc.? Okay, so autotrophic organism and autotrophic further divide into photoautotrophs, chemoautotrophs. Heterotrophic organism can further divide into saprophytic, parasitic. Saprophytic means obtain nutrient from dead or live organism.
Organic matter.
Obtain nutrients, that means organic matter, from dead organism or live organism. From dead organism. That time the nutrition, the type of nutrition, mode of nutrition is called saprophytic. So saprophytic organisms are those organisms that obtain nutrient from dead. Nutrient means organic matter. So many saprophytic organisms also considered as decomposer because they decompose the organic matter, dead organic matter into simple form. Yes or no? They are also called decomposer. And some are parasitic. This is another mode of heterotrophic nutrition. Parasitic means they obtain nutrient from live plants, live animals, live organisms. Nutrient from live organism. So clearly, microbial world are hugely, highly staggering diversity shown in terms of three-part prokaryotes. They can be eukaryotes. Some are acellular, majority are cellular, and they also show diversity in terms of nutrition also. So clearly, in order to study the microbes, we need to classify the microbes, we need to classify the organisms into proper suitable type, yes or no? To study the huge diversity of the microbes, huge diversity of microbes, what do we need? We need to classification. We need classification. And the classification simple language means grouping. Simple language means grouping of living organisms. পার্টে পার্টে ভেঙে দেওয়া। তুমি যদি আজকে জাস্ট ভাবো স্কুল লেভেলে ভাবো, চলে যাও, তুমি কি ক্লাস 5 এর স্টুডেন্টকে ক্লাস 11 এর স্টুডেন্টের সাথে একসাথে মার্চ করতে পারবে? Is it okay to merge class 5 students and the class 11 students or 12 students into a same group? No. Should be there a different group in order to compete in sports as well as in education. In order to compete any time, we need to classify the students into separate group. Just like that, there is a huge diversity. We cannot put all the microbes in a single group. We should classify into proper way by means of structure, inside structure, outside structure, and various ways. We have to be classified. Yes or no? So that's called classification. That means properly grouped. Organism grouped into proper suitable way. Okay, so many scientists tried classification system, and the properly start by the Aristotle. So many scientists start to classify the organism. হ্যাঁ স্টার্টিং স্টার্টিং মানে কি উনিও ওই দলেই ছিল এভারজেন্স থিওরি so many scientists start to classify the organisms. First, in scientific way, start the classification by Aristotle. Then Aristotle classification system slightly modified in a better way by Carolus Linnaeus. And then the scientist three, which is Haeckel, came and modified the Linnaeus classification system, followed by Copeland, and number 5, R.H.Whittaker. Previous Aristotle, there is also some classification is given, but more scientific basis of classification start with the Aristotle system, and Aristotle classified the total living organisms into plants and animals. He classified into total living organism into plants and animals. And Carolus Linnaeus, he came and just modified the Aristotle system into two kingdom classification system. Two kingdom classification system. He also classified into kingdom plante, and kingdom animalia. And the Carolus Linnaeus classification system is very basic. This system very basic and based on gross morphology and some other factor. Based on basically he take two or three character. Number 1, cell wall, present or absent. Just look for that. Number 2, locomotion, present or absent. Number 3, response to external stimuli. Response to external stimuli. Present or absent. So Carolus Linnaeus, based on two or three characters, he classified the total living organism into two kingdom plant and kingdom animal. Now we say that kingdom plantae have cell wall present or absent. Kingdom plant cell wall present. Locomotion present or absent. Absent. And external stimuli, response to external stimuli, plant. হ্যাঁ, রেসপন্স টু এক্সটার্নাল স্টিমুলি, মানে যদি কোনো স্টিমুলি করে কোনো চেঞ্জ করবে, মানে কুইক কি চেঞ্জ করবে, টাইম লাগবে, ইয়েস রেসপন্স করে, বাট দ্যাট টাইম লিনিয়ার did not achieve that response is actually responded by the plant, so no, absent plant. Kingdom animalia, absent cell wall, locomotion present, and external present stimuli, response to external stimuli present. So clearly, any organism that has cell wall, where do we put kingdom animalia, kingdom plant? Kingdom plant. Any organism which have ability to locomote, so kingdom animalia. So by that way, he just arranged the all living organisms into two kingdoms. Clearly, but this system is very basic, just taken 2 or 3 characters. So there are many disadvantages of two kingdom system. There are many disadvantages of two kingdom system, and many organisms actually show both systemic features, both kingdom features. Disadvantage. And disadvantage number 1 mainly showed by the bacteria, because bacteria has cell wall. Yes or no? So if the cell wall is present, what are we going to put? Plant. Bacteria has flagella that has the ability to locomote. So clearly bacteria has two features, animal like features as well as plant like features. So major disadvantage. And some other organism also showed that two features, plant and kingdom like features. Look another example, Euglena, which is a protist. Euglena has flagella. That means it has the characters like animal. And Euglena can photosynthesis ability. So it is a plant-like feature. So like many those organisms, after two kingdom classification, the major disadvantage, many organisms show two characteristics, both kingdom characteristics. So this kingdom system, two kingdom system must be changed because major disadvantage is there. Okay, and changed by Haeckel. And Haeckel changed two kingdom system into three kingdom system. Change two kingdom system into three kingdom system. Three kingdoms now. One kingdom extra added. The added kingdom is kingdom protista. Kingdom protista. And kingdom plant and kingdom animalia still there. The new kingdom, Kingdom Protista, he added the member of algae, fungi, protozoa, and bacteria, which create the major issue, that means two feature characters, and these groups are separately kingdom into Kingdom Protista. So he devised a separate kingdom, Kingdom Protista, and include those organisms which are two features character. Clear? But still there is a problem. Where?
স্যার স্যার ভিডিওটা অফ হয়ে গেছে, ভিডিও কিছু দেখতে পাচ্ছি না।
Video off. Video to on.
আমার মানে কিছু দেখতে পাচ্ছি না, খালি মানে ইয়েটা লোগোটা।
বাকিদেরও কি সেম ইস্যু?
না স্যার।
না স্যার।
ওকে, তো তোমার প্রবলেম একটু চেক করো, নেটওয়ার্ক চেক করো।
আচ্ছা ঠিক আছে স্যার, আমি লিভ করে জয়েন করছি।
হ্যাঁ হ্যাঁ। So Kingdom Protista contain algae, fungi, protozoa, bacteria. This grouping is satisfied all the character, or there is also scope to change into another kingdom system. Scope. Why? Because see, the bacteria and the algae, fungi, protozoa, bacteria is completely prokaryote system, and algae, fungi, protozoa, they are eukaryote system. Number one issue. And number two issue, bacteria unicellular, algae, fungi, protozoa, some are unicellular but majority are multicellular. So no solve of unicellularity, multicellularity, as well as prokaryotes and eukaryotes. So the fourth classification system is came and done by Copeland, and Copeland classified the organism into four kingdom classification. And he created new kingdom, which is Kingdom Monera. Kingdom Monera. And changed the slight name of the previous kingdom. Kingdom Protista changed the name with Kingdom Protikista. Kingdom Protikista. Plant Kingdom plant changed into Kingdom Metaphyta. Change the name of Kingdom plant into Kingdom Metaphyta. And the fourth kingdom, Kingdom Animalia, changed the name with Kingdom Metazoa. Just change the name slightly, but overall features is same. He included all the bacteria into separate kingdom, which is called Kingdom Monera. He included the member of bacteria here. But still, Protista contain algae, fungi, protozoa. Yes or no? Bacteria created problem like prokaryote, eukaryotic controversy, as well as unicellular, multicellular controversy. Just remove the bacteria and enter into give the separate kingdom, that's called Kingdom Monera. Is their problem is solved, or still there is a scope of improvement? Problem is solved. Copeland just change, introduce Monera and introduce and enter the bacteria, sole bacteria member into that. Problem is solved, or still is there? Still problem is there. Still problem is there because now fungi create main problem. Fungi create main problem, major issue, because fungi mode of nutrition is totally saprophytic. Totally saprophytic means heterotrophic mode of nutrition, whereas protozoa, algae, majority algae, most of the algae, and mainly 100% algae are autotrophic. They can do photosynthesis. As well as protozoa, in category of protozoa, just like Euglena. Euglena has the photosynthetic ability. They had chloroplast. So now the problem creator is the fungi, because fungi is completely heterotrophic and other are auto as well as hetero. Clear? So Robert Whittaker, R.H. Whittaker introduced five kingdom system. Introduced five kingdom system. And he created which kingdom? Fungi. Five kingdom system, total five kingdoms are Monera, kingdom Monera, kingdom fungi separately allocate the fungi into separate kingdom fungi, and change the name. Previously that present in the Haeckel group, Protista, plant and animal. So these are five kingdom of Robert Whittaker proposal. So he created only kingdom fungi, not the Monera. Remember that. Yes, Monera is created by Copeland, not the Robert Whittaker. Clear? Robert Whittaker just introduced fungi. The problem creator is fungi after the Copeland four kingdom system. So he just remove the fungi and enter into a separate kingdom fungi. Monera is still there. Monera is created by the Copeland, not by the Robert Whittaker. It can be a question, short question. Who created kingdom Monera? And majority answer will be Robert Whittaker, and answer is wrong. Monera kingdom created by Copeland. Whittaker just introduced fungi. Monera is still there. So that is the improvement of classification of living organisms. Started with Aristotle and end with five, not end. And it is five kingdom system, more compacted system and well accepted system. Five kingdom model, more accepted system. But this system also have chance of improvement. Why? Because the improvement in the field of two field, improvement in the field of molecular biology, biochemistry, mainly molecular biology and biochemistry also improved that time. And these two fields encourage the change of five kingdom system from three domain system. After the huge study of molecular biology, improvement of the molecular biology, the five kingdom system further changed into now the kingdom. The word kingdom is changed with three domain model or system. So five kingdom system, it is accepted system. Still we are now used but most modern way to classify the organism based on use of molecular biology, use the technique of molecular biology, not the morphology, internal structure, etc., etc. Robert Whittaker used variety of criteria, but still no molecular biology is used. But that time, because that time no molecular biology concept is present, so Robert Whittaker used criteria for Robert Whittaker. Next topic. Criteria for Robert Whittaker Five Kingdom system. হ্যাঁ molecular biology তারপরে developed হয় and scientists believe now that molecular biology is the most important tool to classify any organism. Okay, so that's why three domain system is more modernized and it is now well accepted. Robert Whittaker system is also accepted before molecular biology establishment, and the criteria for he used some criteria for five kingdom system. Five kingdom system. I'll make a comparison list. Characters, five kingdoms are Monera, fungi, protista, plant, animalia. So you use character or criteria number 1, cell type. So first you check which type of cell is the identified organism is which type of cell. Monera is prokaryote or eukaryote. Cell type means prokaryote or eukaryote. Cell type means you just check the organism is pro or eu. Prokaryote is Monera is prokaryote or eukaryote because all members are bacteria, so it is completely prokaryote system. Fungi, pro or eu?
Eukaryotic, sir.
Fungi completely eukaryotic. Fungi eukaryotic system. Protista? No, no. Eu. Plant? Animalia? So only Monera is prokaryote. Only bacteria is prokaryote and rest of the all kingdoms are eukaryotic organism. Rest of the all organism, fungi, protista, plant, animalia, all are eukaryotic organism. Characters number 2. Body organization. Body organization. That means the body is composed of single cell or multiple cell, unicellular or multicellular. Unicellular or multicellular. Answer. Monera is uni or multi? Unicellular. Fungi is uni or multi? Fungi uni.
Multicellular.
Fungi multi, but some exception is there, that is yeast. Yeast is only the exception where it is unicellular. Protista, uni or multi? Answer. Protista unicellular or multicellular? Unicellular, completely unicellular. These are organisms, they are eukaryote, but they unicellular organism. Eukaryote unicellular organism is Protista kingdom. Plant multicellular, obviously multicellular. Nuclear membrane, present or absent. Based on that, we classify present or absent. Now answer me. Monera, nuclear membrane is present or absent? In Monera kingdom, a nuclear membrane, present or absent?
Absent, sir.
Absent. Correct. Because the prokaryote organism is named so that here the nucleus is not well structured. Nuclear membrane is absent. So it is a prokaryote organism. So nuclear membrane, same. And rest of them all are eukaryotes. That means all are present.
Present.
Number 4, mode of nutrition. Another character we take, mode of nutrition. Autotrophs or heterotrophs? Answer. Monera. Auto or hetero or mixed? Not only auto, hetero also there. Some bacteria can infect in your body, so they are hetero, mixed, both types. Diversity, well diverse, both. Fungi auto or hetero? Fungi auto na hetero? Why Whittaker introduced separate kingdom fungi? Because fungi main creator problem. I previously said that fungi is totally heterotroph, is totally saprophytic. That's why we need to introduce a separate kingdom for the fungi. Otherwise, the discrepancy is there because protozoa, algae, they are autotrophic, but fungi is completely heterotrophic. So how can you two organisms introduced in the same group? So there is a discrepancy. That's why Robert Whittaker introduced into the separate kingdom. So it is hetero, completely hetero. Fungi is completely hetero. No hetero autotrophic fungi is there. Protista, auto or hetero, both. Both. Plant completely auto. Animalia completely hetero. Number 5, cell wall structure. Hmm? Cell wall structure. Cellulosic or non-cellulosic? Cell wall structure, cellulosic means made up of cellulose, or non-cellulosic means made up of other component other than cellulose component. Answer. Monera cell wall composed of cellulosic or non-cellulosic material? Non-cellulosic, it is a peptide of glycan. Correct. Non-cellulosic. Fungi, fungi Cellulosic or non-cellulosic? Non-cellulosic bacteria, Monera kingdom are composed of peptide of glycan, CN. And fungi cell wall structure, it is also non-cellulosic. You know the name, that's
Sir,
called chitin. So chitin, not cellulosic, it is a non-cellulosic chitin. So chitin is non-cellulosic material that made of the fungal cell wall, non-cellulosic. Protista, cellulosic or non-cellulosic? No, non-cellulosic. Non-cellulosic. Silica and other components made of their cell wall. Plant, cellulosic. Animalia absent, no cell wall. So these are some characters taken by the Robert Whittaker to classify into five kingdom system, and we compare that with five kingdom. But we said earlier that this kingdom system, this five kingdom system is not final. It is also improved after that when molecular biology established, molecular biology improved, molecular biology techniques are improved. That time, this five kingdom system changed with domain system and called three domain system. Correct? So next topic, three domain system. Three domain system introduced by Carl Woese in 1977. That means recently, more recent. Robert Whittaker work at 18th century. It is the 19th century. So three domain system introduced by Carl Woese in 1977, and he proposed domain over kingdom. Over kingdom. So before Carlos, the all classification system, the suffix is kingdom, two kingdom, three kingdom, four kingdom, five kingdom. The kingdom is the highest taxonomic category there, but Carlos introduced the domain is the highest category, taxonomic category, and the kingdom is replaced with a domain. And he showed that there is three domains. That's why the term is three domain system. He classified the organism into three domains. He classify the organism into three domains. And the three domains are domain archaea, and domain eukarya. So he classified the total organism into all living system into three domain: domain eubacteria, domain archaea, and domain eukarya. Domain eubacteria, domain eukarya. Actually, these two are the bacterial group, but separate into two different domain. These two are the separation of or splitting of Monera kingdom. Monera kingdom, that is previously I said that this kingdom is solely composed of bacterial member. This bacterial member he classified into two different domain. One is archibacteria, that means domain archaea, and other is domain eubacteria, that means modern bacteria. Archibacteria means primitive bacteria, and domain eubacteria contain modern bacteria, true bacteria, etc. So he split the domain, kingdom Monera into two domains. So it is part of kingdom Monera part, domain Archaea and domain Eubacteria. And rest of the all kingdoms he included into domain Eukarya. Other kingdoms are in five kingdom: kingdom fungi, protista, protista, animalia, and plant. Because they all are eukaryote, yes or no? Kingdom fungi, protista, animalia, plant, they are all eukaryote. That's why he separately a domain into a domain eukarya. See the name eukarya means this domain contain all eukaryotic organism. So other four kingdoms, that means the fungi, protista, animalia, plant, they are all included into a single domain, which is domain eukarya. But the kingdom Monera is split into the two different domain. One is archibacteria domain, domain archaea. Other is eubacterial domain, which is domain eubacteria. What is the basis of three domain system of classification? Basis. I previously said that it is a completely work of molecular biology, and Carl Lewis checked the sequencing of to classify the organism. He just checked the sequence of rRNA, small subunit, simply rRNA. So he do not look for the morphology. He do not look for the internal structure, cell wall, locomotion, etc., etc. He just check the sequence of rRNA structure. Any organism he identified, check the rRNA structure. rRNA can be of two types. One is 16S rRNA. Can be a variety of type. 16S rRNA, he look for only 16S rRNA and 18S rRNA. That means rRNA he taken for a sequencing is small subunit of ribosome. Did you know the full form of rRNA? R means small r means in our cell RNA can be of three types, simply M, T, and R. Carl Woese look for rRNA, not M or T. What is the full name of rRNA? The prefix R stands for
Ribosomal RNA.
ribosomal RNA. So these rRNA found in ribosome. Found in ribosome. So rRNA means ribosomal RNA. That's why the rRNA found in ribosome. And he choose that ribosome has two subunits. One is small, other is large, and he choose small subunit for working. He choose small subunit for working. And clearly, when he choose to sequence a small subunit of rRNA, so there is two types. One is 16S and other is 18S rRNA. 16S rRNA and other is 18S rRNA. And he clearly found that 16S rRNA found in all prokaryotic organisms. All prokaryotic organisms. And 18S rRNA found in eukaryotic organism. Eukaryotic ribosome contain 18S rRNA and prokaryotic ribosome small subunit contain 16S rRNA. So clearly the three domain model, this domain eukarya contains 16S or 18S rRNA? 18S rRNA. And domain eubacteria and domain archaea, as they are prokaryote system, they contain 16S RNA. Clear? But in prokaryote system, the 16S rRNA can be comparable. Means eubacteria and archaebacteria, there is sequential difference. Sequential difference in the 16S rRNA. Just check for this difference and you can easily compare. After the comparing, you can easily identify which types of prokaryotic is this. It is a domain eubacteria or domain archaea. That means this 16S rRNA can be further comparable in the prokaryote. This 16S rRNA sequence, by sequence comparison, 16S rRNA can be of two types. One is eubacterial type, other is archaebacterial type. One is eubacterial type, 16S rRNA. Other is archaebacterial type, 16S rRNA. So by that, he creating two domain. One is domain eubacteria, other is domain archaebacteria. And other organisms are all containing 18S, no 16S rRNA is placed into the domain eukarya. You take fungi, protista, plant, animal, any organism from that show not 16S, it is 18S. So created a different domain, that is domain eukarya. Three domain model classification basis clear or not? Understood? একদম completely কোনো internal outer structure না, just identify the organism and check for the ribosome, mainly small subunit ribosome, and take 16S RNA. Small subunit ribosome also contains 16S 5S RNA. Clear? Other RNA also present, but he choose 16S RNA because of different reason. 16S RNA is highly stable, can do not change over the time. That's why he takes 16S RNA over other RNA molecule, rRNA molecule. So he choose 16S RNA from the small subunit and just sequence. If it is 18S, placed into the domain eukarya. If it is 16S, now check for the sequence comparison. And after this comparison, you can find it is a eubacterial type or archaebacterial type. And by that way, you can put the any organism into distinct domain. হ্যাঁ 16S এ যখন তোমার সিকোয়েন্স কম্পেয়ার করবে, একটা ইউব্যাকটেরিয়া যদি আসে তার সিকোয়েন্সের প্যাটার্ন চেঞ্জ হবে, আর আর্কিব্যাকটেরিয়া সিকোয়েন্সের প্যাটার্ন চেঞ্জ হবে. বাই লুকিং ইনটু দ্যাট ইউ ক্যান প্লেস হুইচ টাইপ অফ rRNA it is. If it is eubacterial type 16S rRNA, that is a special sequence, some signature sequence is there, and some by looking signature sequence you can find out which type of rRNA it is. It is eubacterial or archaebacterial. and placed into that organism. So more simple way to classify the organism is Carl Woese. Rather took cell wall, but Robert Whittaker did. He take 5 or 6 characters to check and confirm which category they belong. But it is very simple. By using molecular biology technique, it is very simple to identify organism that you identified which domain it belongs. If it is 18S, eukarya. If it is 16S, now compare that sequence. If it is eubacterial type, placed into domain eubacteria. Archibacterial type, placed domain archaea. So from whose concept, the Monera kingdom further can be divided. Further can be divided because of the 16S rRNA comparison analysis. Bacteria can be of 2 types. One is majorly divided into based on the rRNA sequence. One is eubacteria, other is archaebacteria. Clear? Online students, 3 domain system classification.
Yes, sir, clear.
Yes, sir.
Now compare this domain system with some characters, which is heavily important for question purpose. Three domain system comparison. Characters, domain, eubacteria, domain, archaebacteria, and domain eukarya. First character, prokaryotic cell structure. Okay, cell structure only. Means prokaryote or eukaryote? Tell me. Eubacteria, prokaryote or eukaryote? Prokaryote. Good. Archaea, domain archaea? No. It is from the Monera system, Monera kingdom. That means it is prokaryote. And eukaryote, its name suggests it is eukaryote. Clear? Second character, membrane enclosed nucleus with nucleolus. That means proper structure of nucleus present in domain eubacteria, yes or no? Proper structure of nucleus present in eubacteria, yes or no? No, because it is prokaryote, so no membrane enclosed nucleus and nucleolus. Archaea also no, but eukarya. Point number 3, internal membranous organelles. Internal membranous organelles. Try to answer, online students. Internal membranous organelles, domain eubacteria, present or absent, yes or no? Eubacteria contain internal membranous organelle? No. Archaea? Eukarya?
Yes.
Yes. Cell wall. Eubacteria contain, what I said previously, eubacteria contain
Cellular
non
present has new bacteria.
non-cellulosic means peptidoglycan. Peptidoglycan. Or murin. Another name is murin. But the domain archaea has the same cell wall structure. So what changed? The name is changed from the peptidoglycan and is given pseudopeptidoglycan or pseudomurin. The cell wall structure present in archaea is pseudopeptidoglycan or pseudomurin. And domain eukarya, various type. It can be chitin, it can be cellulose, sometimes no. So various types. So but no murin is there. No murin is there. The comparable is no murin. Other than murin, cell wall composition can be. Clear? Number 5. Membrane lipids. Important comparable feature. Membrane lipids. Eubacteria. The membrane lipids are ester linked. Membrane lipids are ester linked, straight chain, and contain fatty acid. Straight chain fatty acid. How the lipids composed? Lipids composed of glycerol and fatty acid. Okay, so glycerol and fatty acid, the connection is ester or ether. Ester linkage occurs. The connection between the lipids, two molecules, glycerol and fatty acid, the connection, the bond involved is ester link. So ester link found in eubacteria, and normal fatty acid, straight chain fatty acid, that means oleic acid, palmitic acid, etc., saturated or unsaturated fatty acid normally found clear in the cytoplasmic membrane of eubacteria. But in case of archaebacteria, the linkage is different. The linkage is different, and the linkage is ether, not the ester. Important point. The cytoplasmic membrane, the structure is composed of ether link in archaebacteria, and there is no proper fatty acid chain. It contains branched isoprene derived chain. Important point of difference between archaebacteria and eubacteria, cell wall also are differ, as well as cytoplasmic membrane also differ. Cytoplasmic membrane, how that difference differ in linkage as well as the fatty acid composition. Here is it is actually the fatty acid and it is straight chain fatty acid, but no trace of fatty acid, but composed of isoprene derived large unit, and it is branched unit. There is no branch. Fatty acid is straight chain, that means no branching. But here branched as well as isoprene derived unit, no complete fatty acid is there. Point clear? Point clear? And domain eukarya show the structure of membrane lipids like eubacteria. That means sterling and stretch chain fatty acid. This point is clear or any doubt? Membrane lipid.
এটা কি মানে ডোমেন ইউক্যারিওট সবার মধ্যে স্ট্রেস লিংক
একদম all from fungi to human. This point is clear to all.
Yes, sir, clear.
Okay,
স্যার একটা ডাউট আছে
বলো।
স্যার আইসোপ্রিন মানেটা কি?
আইসোপ্রিন মানে হচ্ছে লং ইউনিট অফ হাইড্রোকার্বন, লং ইউনিট অফ হাইড্রোকার্বন কম্পোজড অফ কমপ্লিটলি হাইড্রোকার্বন।
ঠিক আছে
ঠিক আছে, that means fatty acid না। fatty acid, the structure of simple fatty acid are COOH, that means a COOH group থাকবে। Clear? তো এই স্ট্রাকচার তুমি এখানে পাবে না আর্কি ব্যাকটেরিয়া, ও স্ট্রাকচারটা হাইড্রোকার্বন দিয়ে তৈরি এন্ড ডিফারেন্ট অ্যাজ ওয়েল অ্যাজ ব্রাঞ্চ। এখানে হচ্ছে সিম্পল স্ট্রেট চেন থাকে। as well as bonding or differ. Next character point. Gas vacuum. Gas vacuums. Eubacteria present. Gas vacuum is a feature of bacteria. Archaebacteria also present. Domain eukarya absent. Domain eukarya contain vacuole, but not the gas vacuole, gas containing vacuole. Vacuole,
প্ল্যান্টের মধ্যে তো
vacuole থাকে, but the function is different. The content is different. Gas, the content, sorry, vacuole present in the plant contain nutrients, carbohydrate, protein, fat, etc. storage unit, but it acts as a gas. So gas formation, gas vacuole is the part of bacterial feature. That's why eubacteria and archaebacteria both gas vacuole is present, but eukarya absent. Gas vacuole actually creates the buoyancy to float into the aquatic surface. Number 7 point. Chromosome. Circular or linear? সার্কুলার মানে সার্কেল না লিনিয়ার মানে স্ট্রেট?
তাহলে তো এটি সার্কুলার
ইউব্যাকটেরিয়া আর্কিব্যাকটেরিয়া সার্কুলার, the DNA structure, chromosome structure is circular. Archaebacteria also circular, but the domain eukarya has linear structure. Circular means the DNA structure like that, and linear means linear.
প্ল্যান্টের মধ্যে লিনিয়ার
Linear. All eukaryotic organism contain linear chromosome. Next, histone protein, present or absent? Histone protein. What is histone? What is histone protein? And where the histone protein found? What is the basic function of histone?
স্যার ডিএনএ প্যাকেজিং এ দরকার হয় তো।
DNA প্যাকেজিং এ দরকার হয়, মানে নিউক্লিওজোম স্ট্রাকচার ফরমেশন এ দরকার হয়। নিউক্লিওজোম স্ট্রাকচার means it is prokaryotic features or eukaryotic features? Eukaryotic features. So histone is a basic protein that bind with the DNA. DNA is positively charged or negatively charged?
নেগেটিভলি।
পজিটিভ কি করে হয়? DNA ফুল ফর্ম অ্যাসিড বলছো, কি চার্জ হবে? সো নেগেটিভলি চার্জ। দেখো তোমাদের বেসিকের কত প্রবলেম, 11-12 এর বেসিকে কোথায় কোথায় তোমার প্রবলেম ধরে রেখে দিয়েছো, মানে কন্টিনিউ চলে যাচ্ছে। ক্লিয়ার করে এগুলো। সো হিস্টোন ইজ আ বেসিক প্রোটিন দ্যাট বাইন্ড উইথ DNA and help to compact. The structure will be now much more compact and for nucleosome. DNA and compacted to form nucleosome. So nucleosome structure is the feature of eukarya. So clearly histone protein highly present in eukarya. But eubacteria, archaea normally absent, but sometimes rarely. Some histone can be present, but we can also write down absent. Right? Absent. Absent. As it is features of eukaryote, histone protein is absent in prokaryote, means archaea and eu both domain did not contain any histone. Number 9, intron. এটাও শুনেছো না? What is intron? দেখি 11-12 এর কি নলেজ নিয়ে এসেছে। Actually, these characters are testing of 11-12 knowledge. What is intron? Intron or exon? Two words are there, intron and exon. What is intron? না না, ওটা axon, ওটা তো ডেনড্রাইট axon, নিউরনের সাথে কানেক্ট হবে। ইন্ট্রনের আনাদার এর সাথে আরেকটা ওয়ার্ড পড়তে, এক্সন। Tell me, what is intron? Online students, class 11-12 part.
স্যার ইন্ট্রন তো নন ট্রান্সক্রিপ্টিং পার্ট না, যেটা স্লাইসিং করে হাঁটিয়ে দিই আমরা।
আর এক্সন?
এক্সন ট্রান্সক্রিপ্টিং পার্ট, যেটা আমার ট্রান্সক্রিপশনে কাজে আসে।
ট্রান্সক্রিপ্টিং আর কোডিং?
সরি স্যার, কোডিং এ, কারণ ওখানে জিন কোডিং গুলো আছে।
Correct. That means intron is the non-coding sequence, non-coding region. From that, no proteins will be synthesized or no amino acids are synthesized. That region is called intron. So intron is non-coding sequence. Non-coding sequence. ইন্টারনেট সার্চ করছো নাকি? কে বলছে এটা?
না স্যার, আমি বললাম স্যার।
ওকে ওকে, ভালো, গুড। Intron means non-coding sequence from where no protein will be synthesized. So clearly, from the mature, from the RNA part, Intron should be removed, spliced out. The process is called splicing. Important question. What is splicing? Class 11, 12 level. এখানে তো অন্যভাবে পড়বো molecular biology আরো ডিটেইলস যাই হোক so intron should be removed and exons are joined. That means eukaryotic gene structures like that eukaryotic gene structure like alternating unit of intron or exon. Red is exon,
black is
exon 1, exon 2, and the black is intron, intron 1 and intron 2, intron 3. So it is matured or unmatured RNA.
mature.
No, intron is present, na? Non-coding part is present. So it is not matured. It is not matured. It is primary. The RNA is primary RNA. It should be matured. Otherwise, no protein will be synthesized. Yes or no? That's why intron is removed and exons are joined. So finally, after the splicing event, 2 exons are joined, 1 and 2, and introns are removed. Black are removed.
তখন তাহলে দিলাম
এবার এখানে আর রাইবোসোম can easily bind and synthesize, start to synthesize translation. So this is the features of prokaryote and eukaryote. It is the features of eukaryote. It is the features of eukaryote where intron is present in between the exon. So make the gene structure split. This gene structure is called split gene. This gene structure is called split. অনেক information নিয়ে যাচ্ছে মাথায় রাখবে. This gene structure is called split gene where the intron is present. So it is a features of eukaryote. So intron is present in eukaryote, domain eukarya, and eubacteria archaea absent but rarely sometimes can be found in some gene. Rarely found. Majority absent.
switching of
Split gene means হচ্ছে intron exon alternatively present. In between the exon, intron is entered. That's called split gene. ব্যাকটেরিয়া জিনটা কেমন হবে বলো তো? পিওর রেড আসবে, pure red, no intron. But eukaryotic primary gene is black red, black red, black red, mosaic type or split type. Done. Point is clear. Intron. Intron point is clear. Online students.
Yes, sir.
Okay. Next point. Character point. From intron point. Number 10. Splicing, capping, polyadenylation. Splicing, capping, polyadenylation event. Present or absent? In which domain? What is splicing? Splicing means intron removal process. Splicing means intron removal process. Capping means in the 5' end, RNA has 2 ends. One is 5' and another is 3'. In the 5' end, guanine residue cap is present, which give the structural stability. Structural stability of the RNA. And polyadenylation, these events also occur in the, but it is in the 3' end. It is in the 3' end of the RNA, and they also give the structural stability. Enhance translation process. Translation process means the RNA that is synthesized, I said earlier, intron, exon, intron, exon. After that, splicing first occur, where introns are removed, exons are joined. Exon joined, splicing event. And it is the 5' and 3' end. It has 5' and 3' end. In the 5' end, for the beta stability, this RNA stability, for purpose it cell add guanyl residue at the 5' end for the capping, and unusual bonding is there. পরে আমরা details এ দেখব. And the 3' end contain a stretch of adenine residue continuous 200 to 300 residue, and that event is called polyadenylation. Polyadenylation means poly means more, adenylation means more adenine residue. Where it is added, it added in the 3' end. So these all events combinedly occur in prokaryotic RNA or eukaryotic RNA. You can answer now easily. These all events occur in prokaryote or eukaryotic RNA. Eukaryotic RNA, because splicing is a part of eukaryotic RNA to become mature. So these two are the another two events also occur in eukaryotic RNA, absent in prokaryote RNA. That means clearly you can now answer splicing capping polyadenylation event occur in eubacteria or archaebacteria. Both are absent. Only eukarya present. দেখো আমি ইয়েস নো করে বেরিয়ে যেতে পারতাম, হ্যাঁ, কোনো টিচার এত ডিটেলসে বলবে না, আমি তোমাকে ক্লিয়ারলি বলে দিচ্ছি এই পয়েন্টগুলো মিনিং, ইফ ইউ নো দ্য মিনিং ইউ ক্যান আনসার ইজিলি। If you do not know the meaning, because at that level, splicing, capping, polyadenylation, these events, these actually molecular biology part. But I try to answer in a simple way. What is splicing? What is capping? What is polyadenylation? Just single line. And by that way, you can easily answer where it is present, where it is absent. So clearly these events are eukaryotes event. So domain eukarya contain or present this event, and eubacteria archaea absent. Answer is clear. Splicing, capping, polyadenylation event, where it is present and where it is absent. It is absent in eubacteria, archaebacteria means prokaryote model. Clear? Next point, mRNA structure. mRNA structure. mRNA structure can be of two types based on organism. It can be polycystonic or monocystonic. Monocystonic and polycystonic. Do you know the meaning of monocystonic and polycystonic? Mono means
Single.
cystonic means single cystone is present. Cystone means structural gene, structural component, protein synthesis. Cystone means single protein is synthesized from the single RNA. That's called monocystonic RNA. But if one RNA make more than one protein, then what we term?
Poly.
Polycystonic. So based on that, the mRNA structure could be mono or polycystonic. Monocystonic means produce single protein from the single RNA. Single protein. Poly means produce more than one protein from single RNA. Meaning is clear? Monocystonic, polycystonic meaning is clear? Our RNA is red. Our RNA is red color. If this RNA make single protein, then this RNA is called monocystonic. When this RNA make more than one protein, then this RNA is called polycystonic. So what prokaryote belongs monocystonic RNA or polycystonic RNA? What do you think?
Domain RT or bacteria.
Domain Eubacteria archaea, it is a prokaryote model.
Monocystonic.
It show monocystonic or polycystonic. Online student, what do you think? Prokaryote model produce monocystonic or polycystonic RNA?
Monocystonic RNA.
Majority answers are monocystonic. No, it's polycystonic. Why? Because prokaryote means bacteria. Bacteria growth time is very small. Very small generation time. That means it rapidly divides into two cells. So it needs more protein in a small time. Yes or no? More protein in a small time, small generation time. So what gives the advantage polycystonic and monocystonic in that view? See, a single RNA produces more than one protein. That means it clearly saves times. That's why bacteria, majority RNA are polycystonic. But eukaryote, it has enough time to synthesize one from one gene to one protein. So clearly it is monocystonic. So domain eukarya contain monocystonic RNA and archaebacteria and eubacteria, as they are prokaryotes, to save time to make more protein from a simple RNA, they contain polycystonic. 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 Eukarya monocystonic. Point is clear?
Just because of time.
Just because of time and make more protein in small amount of generation time. mRNA structure polycystonic or monocystonic? Point clear? Online students, point clear?
Yes, sir.
The list not ends here, but today's class is end. Next day also we discuss another some characters that compared from eubacteria, archaebacteria, and eukarya.